Welcome back, life science learners, to our final installment for today. Let's continue looking at scientific methodology. Before we went into the break, we spoke about the independent variables, we spoke about the dependent variables, we ended off looking at control variables. I mentioned that it was important for us to be able to have these variables controlled. Essentially, the control variables are factors that you keep constant so that it ensures that your results are fair and they're reliable. So what does that mean? It means that if we can control our variables and keep them constant, we're able to produce an experiment that has a fair test. So let's look at the concept of what a fair test is. So a fair test is an investigation where one variable, which is the independent variable, is changed and all other conditions, meaning the control variables, are kept the same or constant. What is measured is referred to as the dependent variable. Let's try and unpack that. So we often have an experiment where we're testing what the independent variable is. We also need to ensure that we're able to measure some variable which we refer to as the dependent variable, and that talks to the effect of the independent variable. But to ensure that our experiment is fair and consistent and produces the same results, we need to keep those other variables constant. I also need to differentiate between the term control, test, and control variables. So let's go back to our experiment where we looked at why plants grow tall. And so if we assume that light is the reason that plants grow tall, or if it's hormones that's the reason why plants grow tall, we would need to ensure that we have a control test, which is one set that is not exposed to the independent variable. So we often have to make sure that we remove the independent variable from that setup to see if that variable has any effect on the plant, or in this case on that group. So in a control test or in a control group, we often remove the independent variable or the variable that's going to cause the impact or change on the dependent variable. So in a vaccine trial, you will have a group that receive the vaccine. However, there will be a group that does not receive any vaccine, and that's the control group. And we want to see if that group shows any symptoms or any effects of having not received the vaccine. So we want to make sure that everything else is the same, but we remove the independent variable from that group. And we refer to that group as the control group, and that group establishes what the other variables are going to have an impact on them. So it's important for us to keep the control variables constant, but it's also important for us to be able to have a control group, and that's a group that we're able to compare our test results to look at the effect of the independent variable as compared to the group which does not have the independent variable. And hence, it's important for us to be able to have controls and to have a control group. Okay. So, a fair test is a test in which the controls are all but one variable when attempting to answer a scientific question. Only changing one variable allows the person conducting the test to know that no other variable has affected the results of the test. And as I mentioned, that this is the best way to know and isolate the impact of the independent variable, is for you to have a control group as well as having the control variables kept constant. Some textbooks, some teachers refer to that as your fixed variables, and things that are kept constant. Okay, cool. So those will rely or will talk to what we refer to as the reliability and the validity of an experiment. So we often refer to things as being reliable or that they're valid. And reliable means you can count on. Valid means it's true, it's accurate. So let's look at these terms in the context of scientific methodology. So reliability and validity are both about how well a method measures something. So let's look at reliability. It refers to the consistency of a measure, whether the results can be reproduced under the same conditions. So When we talk of something being reliable, if you talk about an appliance being reliable or a brand name being reliable, we often means that that brand or that product gives you the same results year after year, or it gives you consistency in its performance. So you often hear of people saying, well, it's a very reliable brand, or it's a reliable car, and that reliability they refer to as being something that is consistent. It gives you the same value for money on a long-term basis. So in scientific methodology, we need to ensure that our results are reliable. So if I conducted my experiment and I had written down a method, and if I shared those results with another scientist and they've collaborated with me, they are able to conduct the exact same experiment and get the exact same results or similar results. If that's possible, we say that the results are reliable, meaning that it can be replicated to produce the same results. That is what reliability is. So it's a fundamental aspect to having a fair test. What is validity? And if we talk of validity, it basically refers to the accuracy of a measure, whether the results really do represent what they are supposed to measure. So accuracy points to the way in which you produce valid results. So when conducting a scientific method, were you accurate in the quantities that you've measured? Were you accurate in ensuring that the temperature was maintained constant? So what apparatus did you use to ensure that you measured accurately? How did you monitor the process? How did you measure time? So that's accuracy. So accuracy again leads to what we call fair testing and producing reliable and consistent results. So let's look at reliability and validity in a comparison table. So when we look at reliability, we know that in first-hand information and data, have I tested with repetition? If you've tested with repetition, we say that you have now produced reliable results. In secondary information and data, so that means if you're doing research through information and collecting data from different sources, how consistent is that information from other reputable sources? Is the data presented based on repeatable processes? So essentially, if you went and did secondary research, and if you went into the internet and you kind of Googled information around a similar topic, are those experiments producing the same reliable results that you have got? If they do, we say that that information is reliable because we're looking at the reliability of yours and we're linking that to that, and hence it increases the reliability when we talk of secondary information. Let's look at validity in the context of primary or first-hand information and secondary. So does an experiment procedure design actually test the hypothesis that I wanted to? Have all the variables been identified and controlled? And if you answer all these questions correctly, we refer to that as being now a valid experiment producing valid results. We can also look at validity in the context of secondary information. So do the findings relate to the hypothesis or problems? So when you read other information or other published sites, do they relate to your primary information or your experiment? And that is what produces valid results. So how do you know your results are valid? If you compare them to a published journal, and if they produce similar results, we say that this experiment has now produced valid results. Okay. So let's look at the application of this. So Spur Bubbles is about a product that SpongeBob has created. Let's look at this. So Patrick and SpongeBob love to blow bubbles. Don't do we all? Patrick found that some Super Bubble soap at the Sale Mart, the ads claim that the Super Bubble soap will produce bubbles that are twice as big as the bubbles made with the regular bubble soap. Patrick and SpongeBob made 2 samples, okay, of the same solution. One was made with 5 ml of the Super Bubble soap, and the other was made of 5 ml of water. So it was mixed with 5 ml of that and water. The other was made with 5 ml of regular bubble soap and 5 ml of water. So they've created a solution having 5 ml of that and the super bubble soap, and another solution having 5 ml of the regular bubble with 5 ml of water. Patrick and SpongeBob used their favorite bubble wands to blow 10 different bubbles and did their best to measure the diameter of the bubbles. So they measured the diameter of the bubbles. So that's talking to what they measured as they popped on the table. So as these bubbles popped in the table, they produced a little a diameter which they measured. The results are shown in the data table on the next slide. So this information was captured. So here we see the results shown in the table, and here's the super bubble which they had purchased, and here's the regular soap bubbles, and here we look at the diameter of these bubbles which have been recorded along this, and they've attempted to do this for 10 different bubbles, and we can see clearly from this the information is presented and that has been recorded in the table. So let's look at this and try and interpret how this information talks to the aim of what these guys were trying to establish. So what is the independent variable in this investigation, and what is the dependent variable? So if we go back to that slide, and if we were to look at what is the independent variable, remember that these guys were looking at the sale mart, and they picked up types of bubbles. So they were looking at the regular bubble soap, okay, and they were comparing that to the super bubble soap. So clearly, if we were to look at what our soap, so that's the one that we're talking about, so that's the super bubble and the regular bubble soap. So that's going to be the type of soap or the type of bubble would be the independent type of bubble. We said that's the independent variable because that's what we're testing. Okay. So that's the independent variable. We also know that they've got to measure these, and so here you see that they've measured the diameter of the bubbles, and so that we refer to as our dependent variable. So that's our dependent variable. So if we go back to the table, we also see that in the table. So here we see that these, which is often in the table headings, are your independent variable, and we look at that in the table, and we can pick that up from the table as well. And further down, we see that the measurement of the diameter of that bubble when it landed on a flat surface was the dependent variable. So the diameter is the dependent variable as we see illustrated in this table. Cool. So let's move on to the next question. Look at the results in the table and calculate the average diameter for each bubble solution. So here you've got to go and use a calculator to calculate that, and so it means that it's important for us to be able to use information from the data table to calculate exactly the diameters. And in this case, you guys would need to add up the sum of all these 10, and you would need to divide that sum by 10. And so essentially it's the total of these, so you've got to add them up, and you've got to divide by 10, and you've got to do the same for the data collected for the regular soap, and that would also be divided by 10 to produce what we call the average. And why is the average done? Remember that in science we repeat experiments or we calculate the averages, and those averages often refer to reliability or consistency of your results. So it's important for us to be able to calculate averages when carrying out a scientific investigation. So I'm going to leave that to you guys to calculate. We're going to move on swiftly to the next question. What do you think SpongeBob and Patrick's conclusions should be? So remember that when they looked at this experiment, they were looking at which of the two. So if I go back to the aim of the experiment was to determine if this super bubble soap was going to produce larger bubbles than the regular bubble soap. And so if we go to the data table, and if you look at the results, and if you calculate the averages, you will see that the super bubble soap produces an average that is significantly higher or larger than the average produced by the regular bubble soap. So again, when we go to our conclusions, we've got to be able to use information. So here you've got to calculate that, and so you would have got a value, and that value would be an average for that. So I'm going to give you an average. Let's say it was 5.10 centimeters, where the regular was, I'm just guessing this for now, 4.10. We can see that clearly that the average for the super bubble was significantly higher. Okay. Are these results reliable? Why or why not? And many of you will choose to either go, yes, it's reliable, no, it's not. There are many flaws in this design of the experiment, or it was only done for 10 bubbles. So we're not really sure if this was reliable, but I can look at the method. So they've used 5 mLs of both the samples, which was fair. They've diluted them in 5 mLs each, which was again controlled. So from a fairness and reliability, that was valid. They also measured 10 bubbles. So it wasn't a measure of comparing 2 bubbles or 1 from each. They've looked at an average. Yes, it might not be consistent, it might not be enough, but we can increase the number of bubbles to give you more reliable results. So for me, I think that these results are quite reliable in that they're producing significantly good results that we're able to compare and contrast and draw a conclusion. Okay, so folks, we're getting to the end of the session. As we overview what we've done today, we've looked at scientific method, we've looked at writing a hypothesis, we've looked at how we lay out an experiment, we've looked at variables, most importantly independent, dependent, and your control variables. We've also spent some time looking at what a fair test is and how reliability and validity are important to ensuring that results at the end generate reliable information. So from my side, it's been a good session. I'll see you guys soon, where we talk about how to manipulate data in a scientific method. From me over to you guys. Take care. Go well.